So our next panel is going to be led by John Dwyer, who's president of the Global Alzheimer's Platform. And he is going to talk about a second prong of what we're doing with the Davos Initiative, which is biomarkers and new innovations in thinking about how to increase access to timely and accurate diagnosis in clinical trials and potential clinical practice. So, John, thank you for taking this on. Thank you for what you're doing, and good luck with the panel.
Well, thank you, George. A brief introduction. The Global Alzheimer's Platform has the mission to increase the quality, reduce the time, and reduce the cost of Alzheimer's clinical trials and, by extension, clinical practice. We are very pleased and honored to be the partner to the Davos program from the clinical trial perspective. I am very pleased and honored to be moderating this particular panel, because not only do we have real experts in this rapidly emerging field, but we also are at a very good point in time. When it comes to therapies, as we saw in CTAD last week, you can put two neurologists together about the relative merits of therapies and you get three opinions. So there's a great deal of learning and consistency yet to evolve on the therapeutic side. Conversely, there was unfathomable for scientists excitement and confidence in the blood plasma tests that we're going to discuss today of all different stripes and character. So we're looking at something that represents genuine good news, both scientifically and for the world at large. Just a couple comments about context as we begin today, and then we'll introduce our panel. A lot of people don't realize that today there are two relatively new approved therapies in the world characterized as disease-modifying therapies that cover 1.6 billion people. China approved a drug from Green Valley, and in the United States, Biogen was approved for Aduhelm. So the clear and present need has never been higher than to determine how to characterize and assess and diagnose patients to determine their condition and determine whether they are appropriate for the therapies that have already been made available and the therapies that are coming. Similarly, and there's been a great deal of discussion around this, the number of clinical trials for therapeutic indications in the world, especially in the United States, but the world more broadly, has also never been higher. So we have not grown the infrastructure for therapeutic clinical trials in any material way. In fact, it's contracted because of COVID. And yet we need to get more done in less time. And these blood plasma tests that we're going to discuss are centrally important to expediting the recruitment process, better characterizing volunteers in clinical trials, and better improving the results of all kinds of therapeutic trials that are underway. This is good news. So today, I would like to ask the panel to briefly introduce themselves and their organizations and the emerging technologies that they represent, and in Dr. Ip's case, as well, her clinical perspective. And I'm going to begin with my friend Sasha. Sasha, please.
Thanks, John, and big thank you to the organisers of this meeting for inviting me to join the discussion today. So my name's Sasha Boziat, and I'm the Global Medical Lead for Alzheimer's Disease at Roche Diagnostics. More than 50% of people living with dementia have no formal diagnosis. And in Europe, people wait on average of 26 months from symptom onset to diagnosis. But what we see is great geographical variation in the diagnostic pathway and the processes, meaning that many centres are actually doing very well at identifying Alzheimer's disease early and with accuracy. This tends to be specialist centers where biomarkers are used routinely as part of the workup. At Roche Diagnostics, we're on a mission to make those specialist centers the rule rather than the exception. We want to streamline the diagnostic pathway so that all individuals, no matter where they come from or where they live, can have access to an accurate and timely diagnosis. We do this through the provision of CSF tests of A-beta and tau, which are standardized, well-validated, and fully automated, so they're really designed for clinical routine use. But we know that there's still a significant bottleneck at the primary care level. So we're also working on the development of blood-based biomarkers with a view to providing an affordable, accurate, and easy to use referral tool for primary care physicians. I'm really pleased to be here with you all today and to have the opportunity to discuss these points in more detail as we move through the discussion today. Thank you.
Thank you, Sasha. Dr. Joel Brownstein, who also happens to be my spiritual guide. It's good to see you again, Joel.
Good to see you, John. And like Sasha, really appreciate the conference organizers and the honor and privilege to be able to present here today, talk a little bit about what C2N Diagnostics is doing. My name is Joel Brownstein. I'm CEO of C2N Diagnostics. By professional background, I trained in the field of cardiovascular medicine and health policy, and I was able to see the evolution of cardiovascular biomarkers really becoming part of the standard of care, starting as early as the late 80s and really into the 90s, with LDL cholesterol really serving as a guide to help manage preventive risk factors. And with that aggressive risk factor modification came dramatic improvements in cardiovascular outcomes. And so what's really exciting about where we are at both C2N and the field within Alzheimer's is that I almost feel like we're in the early days where we're seeing a transformation in the field that will ultimately enable very broad access of blood-based biomarkers that are highly precise, can be used to guide treatment decisions, can be used to differentially diagnose conditions that may masquerade as Alzheimer's disease from other disorders. And at C2N Diagnostics, we're really involved with all elements of the diagnostic pathway for patients with various forms of neurodegeneration. Our initial product is a product called Precivity AD, which is a blood-based biomarker test that measures apolipoprotein E proteotype, which is equivalent to the genotype, as well as amyloid beta 42 and 40, which are peptide fragments that are highly indicative of what's happening in the brain. And with now data that exceeds over 10,000 patients across a whole variety of independent studies and clinical experience, where we've now introduced the test within the United States, we're seeing a great deal of both precision and the ability to help clinicians better identify and remove the diagnostic certainty associated with Alzheimer's disease. So I'll look forward to talking a little bit more about Precivity AD, how we see the landscape changing, and what else we're doing to advance the diagnostic journey for patients for better identification and treatment.
Thanks, Joel. Doctor, if you would please introduce yourself and please take a moment, if you're so inclined, to just describe the situation as you see it in China.
Thank you, John. I really appreciate the opportunity to join this panel discussion. I'm Nancy Ip, Vice President for Research and Development, the Morningside Professor of Life Science, and the Director of the State Laboratory of Molecular Neuroscience at the Hong Kong University of Science and Technology. My current research focuses on the identification of biomarkers and therapeutic targets for Alzheimer's disease, AD. As an example, my lab has demonstrated the roles of a cytokine, interleukin-33, in alleviating AD pathology by promoting the induction of a beneficial microglial subpopulation to clear A-beta. And we have initiated the preclinical study on the therapeutic use of this cytokine. Among other key efforts, I have led the establishment of multi-omics AD databases in the Chinese population. We have identified novel genetic risk factors for AD through genome-wide association studies of AD patients, as well as discover novel biomarkers for AD by undertaking single-cell transcriptomic analysis and plasma proteomic analysis of AD patients. Through a comprehensive profiling of plasma proteome, comprising of more than 1,000 proteins in AD patients of Chinese population, we have identified a panel of 19 plasma protein biomarkers of AD based on the analysis of plasma proteins that are altered in AD patients compared to age-matched healthy controls. We believe that these findings can potentially lead to the development of a simple and robust blood test with high accuracy for early detection and screening of AD at the population scale. Development of blood tests to identify individuals who have a higher chance of developing AD would allow early detection, early identification of these patients and informed diagnosis. It would also allow us to better stratify the patients for clinical trials. We're now working on identifying blood biomarkers for individuals with mild cognitive impairment. And we do believe that the use of blood biomarkers will not only help to assess the conversion to AD, it would also facilitate the design of clinical trials. I look forward to further discussion later on.
Thank you. Michael, tell us what's going on with you in Philadelphia.
Thank you. So I'm Mike Pontecorvo. I'm VP for Clinical Development at Avid. Avid is a wholly owned affiliate of Eli Lilly. So Avid, many of you may know, is involved in development of PET tracers. We actually developed the first approved amyloid PET tracer and the first approved tau PET tracer. And I'm proud to say I think we've changed the practice of medicine, and we will change the practice of medicine going forward. We've demonstrated that biomarkers can be effective in diagnosis, and I think those PET tracers will play a large part in enabling the development of the new generation of plasma markers. We at Avid and Lilly are also working in the plasma marker field. Again, many of you are familiar with the p-tau217 antibody that we've developed, and we've actually collaborated with some of you on that antibody. Lilly also has a long history of research in therapeutic development for Alzheimer's disease, and has several compounds in late-stage trials. I'll give you two personal notes. I worked at Janssen many years ago, sometime in the last century, when dinosaurs walked the earth, and was actually involved in what may have been the first attempted disease-modifying therapy, a drug called sibeluzole that was supposed to stop tau phosphorylation. And we had some hints in phase 2, and we thought the compound looked good, and we fell just short in phase 3. And the frustrating part of it was we didn't know if it was getting in the brain. We didn't know if it was doing what it was supposed to do. We had no way of knowing whether or not it was changing tau in the brain. And I swore I was never going to study another disease-modifying therapy at that point in time. When Dan Skavronsky called me up and said, hey, I've got these PET tracers, you can see the pathology, I jumped. And that was 15 years ago. And we had a vision of changing the world such that diagnosis would be based on molecular pathology, and treatment would be targeted at molecular pathology. And we're getting there. We're very close to that right now. One other personal note, when we first published or tried to publish our first studies, they were rejected because the people who reviewed them said, You must be doing something wrong because 30% of your Alzheimer's patients are negative on your amyloid scan. Well, guess what? We weren't wrong. The overwhelming pattern these days is anywhere from 15 to 50% of patients who come with a clinical diagnosis of AD or MCI-AD do not have amyloid pathology, do not have Alzheimer's disease, and yet they're getting this diagnosis. And that is just unacceptable. That's a life-changing diagnosis. And to have that life-changing diagnosis when we have molecular markers that would correct that is just wrong. So this is our passion, and all of us here on this panel, I think, share that passion.
Not last but not least, while Dr. Ip wins the award for being up the latest, Steve Verduner gets the award for being up the earliest. So Steve from California, tell us how you're evolving and your perspective on the market.
Well, thanks, John, and thanks to the organizers. It's a privilege to participate here today and very much appreciated. So I'm Steve Verduner. I'm CEO and co-founder of Neurovision. Neurovision is a company that's developing biomarkers and diagnostic solutions for the neurodegenerative space, obviously with a focus on Alzheimer's, but we also have plans for Parkinson's and pipeline for multiple sclerosis as well. My background, very briefly, is really coming up through ophthalmology from the early days. I think the lessons learned there were that when we have the tools to measure, then we can get breakthroughs in treatments. And in ophthalmology, we have breakthroughs in structure, measuring function. So we get new treatments for macular degeneration and diabetic retinopathy. And I think we're right at the cusp of that here when we look at what's happening in neuroscience with respect to cognition, blood biomarkers, and imaging and other modalities. We're right there, ready for these aspects to inform on new treatments, to enable and empower new treatments. There's two main aspects of our business. One is retinal imaging, which we're not going to talk about today with respect to detection of amyloid in the retina, but also importantly, vascular risk factors for mixed dementias and cerebral amyloid angiopathy. But really the focus of today on our blood biomarker business, where we have a very unique platform that has ultra high sensitivity and can practically multiplex 10 to 20 biomarkers to create a biosignature, and it's highly scalable. Initial tests are a proprietary amyloid test targeted predominantly at primary care physicians with high negative predictive power, so that we can really rule out true negatives and gate those people through to primary care, to neurologists and to amyloid PET and CSF. In that test, we're achieving very nice performance of our work in progress area under the curve of 0.8 to 0.9. So that's going in a good direction. Also for neurology, more so for neurology, we're creating a multiplex. And currently we have running from a single small blood sample where we're looking at intrinsic amyloid beta 42 and 40, p-tau 181, 231, 217, GFAP, total tau, and neurofilament light. Sort of like an ATN chip, as we say. It's a magnetic particle-based technology similar to classical ELISA, but with a detection scheme based on magnetic particles. Technology ultimately was Nobel Prize winning out of Stanford several years ago. So we're embracing a combination biomarker approach, and we hope to bring other future targets to the platform like TDP43 and others, for which we've got antibodies in the freezer, which we'll get to at a future point in time. I think the other thing that's of note, and this is all on track to launch as a laboratory developed test in 2022. So this is near term. First the test for primary care, and later in 2022, multiplex for neurology. Another aspect for non-high resource countries is a point of care version of this. So again, it's not high throughput, but again, in countries that just don't necessarily have access to this technology, we'll work on a solution for that as well. And so that's in development, up and running on more traditional biomarkers, which we're looking to migrate to some of our targets on the higher throughput system. It's also, the high throughput system is very highly scalable through partners, which is absolutely positively key to this. So finally, I think, blood biomarkers really are the key to enable and empower new treatments and to enhance. And I think, where we'd like to see things go is the pharmaceutical industry really embrace this further, so we have more of a precision medicine approach. So thank you. I look forward to sharing more in the discussion.
That was great. really thank the panel for their perspectives. What I think that we should begin with is what is less often talked about in academic / clinical scientific discussions is scaling. Let's begin with how are we going to bring your respective technologies to billions of people, and what does that entail? And I'd like to ask Dr. Ip first to talk to us about what is success in China for scaling the kind of solutions represented here and introducing them to your countrymen across from the rural to the cities.
So in China, actually, there are difficulties, right, in popularizing the PET scan and also CSF tests. It is very difficult to convince the patients to do the CSF test because of the cultural difference. So I do believe that the blood test will be welcome in China. And there are different ways to implement the test. I think one way is to do it at the hospital. So there's the diagnostic department at the hospital where if you have CFDA approved equipment and also diagnostic reagents, then the test can be done similar to a routine blood test in the hospital. Or alternatively, we can consider establishing service center for the blood test. And so in mainland China, blood-based biomarker tests can be implemented in a third-party service center outside the hospital. So in terms of implementation, it can be done. Now in China, currently a lot of the diagnosis in the rural area sometimes is not very accurate because in the rural areas, they usually rely on interns that have little experience, and so it can result in misdiagnosis, inaccurate diagnosis, and also misdiagnosis. So I think this is a problem in the rural areas. In the urban areas, I think is better because there are specialists, even though the number is still insufficient, but the diagnosis can be made in the medical universities, or it can be made in the hospitals. Still, the cognitive assessment mainly performed using MoCA and MMSE. So doing PET scan and also CSF tests is just not the norm in China. So I do believe that if we have blood tests that are sensitive and accurate, it will be welcome in China, and it definitely would provide a much better alternative than simply doing the MoCA test or the MMSE, because in China to have PET scan and to have routine CSF test is just not practical.
So, Sasha, the ELISA platform of Roche DX is well known. It's highly distributed, but this is a different application. We've got doctors that have little or no frame of reference for the blood tests that we're talking about. What's your company's perspective on doing this in scale and bringing it to the world in a accelerated fashion?
Yeah, well, absolutely, John. I mean, yes, the Alexis platform has got a global footprint, and the scalability is something that we've really embraced with our CSF tests. And we've done a lot of work to, ensure that we've got a very simple and standardized protocol, and that we get very little variance. And this is something that can really be implemented across clinical routine, across different cobas machines. So this is something that we're well versed in, let's say. But yes, of course, as you say, blood tests, this is, a new innovation really, and bringing it also into a new point in the diagnostic pathway. So I think, we're having a lot of discussions now around, how we actually educate and upskill primary care physicians in order to be able to clinically accept and adopt these tests into their clinical routine, let's say. And I think really the key thing is also, to consider in the development process is that really we need to provide something which gives a very, clear and straightforward answer as to refer on or not. So I think any kind of predictive algorithms, any kind of risk scores are going to be very difficult for primary care physicians to be able to understand and most importantly to communicate to their patients as well. So we're looking to try to develop something which really gives a very simple answer to be used alongside a cognitive test. To your point, Nancy, these individuals are coming in with a cognitive complaint. So this needs to be investigated. We need to respect that. And I think it will be the combination of a cognitive test and a simple blood test that will hopefully equip a GP to be able to make that decision to refer on or not. And we don't want to, give them really any more responsibility than that, because they already have, an awful lot to deal with. But there's a lot of education around that. And, this is something we're starting to look at now.
Mike, what's your perspective on this? You're particularly engaged with 217. How do you see it rolling out in clinical practice?
Right.
And it's and the story.
So I don't want to minimize the difficulties. It is not trivial to take an assay like this, like any of them we're talking about, and develop them. I know Sasha Roche has spent years building the electrocyte platform, and to roll out a blood plasma assay in months is really a high bar. And so our number one point will be to make sure that we've got it absolutely nailed down, correct and reliable, time after time, and using bots and all the technical work that needs to be done, and have it solid in our lab before we even think about putting together a test that can be exported to partners or point of care or anything like that. And that's a pretty big technical challenge, and it's going to take some time. It's not going to happen in the next couple of months. I mean, we hope to have our lab up and running sometime in 2022, but to go beyond that, I think would be a real challenge. I think the other thing we have to keep in mind is we're still learning about these various blood assays. It's still the case that where it's available, a PET scan probably gives you a more definitive answer than a blood scan or a blood assay, although a very high value on a blood assay is almost certainly going to be associated with Alzheimer's pathology. A very low value inverse for the A beta is going to be associated with absence of pathology, but we're seeing an in-between segment that's going to be harder to diagnose, and some of that may be the biology. That patients don't flip a switch and automatically go from no pathology to a ton of pathology overnight. And some of it may be the particular characteristics of the assays where they really catch very early stages of disease, but maybe stages that won't progress very rapidly. And we need a lot of research still to understand that. So I think we need to mix realism with optimism. I think these new plasma tests provide a tremendous amount of potential, and I think they're going to be available at one level or another in the near future. But I think we also have to realize there's still an awful lot to learn. I mean, look at how much we learned from the PET scans, how much we learned about the disease and the order of changes in the disease. And I think having plasma assays available on a wide scale, we're going to learn even more.
So Joel, listening to this, I really like your and Steve's point of view on the scaling question, because as a patient advocate, having had the same experience that Mike mentioned with my own family, we're right now at about a 30% misdiagnosis rate in my large family. I'm of the view that we should, and this is a little bit of heresy perhaps, but I'm of the view that you don't make better the enemy of the good, that getting a blood plasma test in the marketplace today of some level of accuracy, sensitivity, specificity way outweighs learning more because of the current condition of misdiagnosis and the costs of the very important PET tests. I'd like C2N and Steve's view of these things.
Steve, would you like to go first, or you want me to? Why don't you go first?
Go ahead, Joel, and you want me to go first? Okay. Well,
I'll be happy to. I hate to see you go last the second time around. So.
Okay, you want me to go? Sorry.
Sure.
Yeah, sure. So I think these are all really good points, and I think that we all have to be mindful of being responsible in how we bring tests to the market. And so, in our company, people are like, well, what have you been doing these last several months? All these, design of experiments and looking at every aspect of what we do, which those who go to the details know what that's involved, and improving our precision to get good coefficient of variation, temperature, humidity, pH, buffer composition. The devil is in the details. And so it is an extensive effort. That's what we've been doing with our time before we start running the next group of high value samples. So whatever we bring forth, I think that we have to have many of them initially a laboratory developed test pathway, which for the most part is not very regulated, except for the CLIA lab. We need to be mindful and have an FDA type of mindset. And what I mean by that is, that's why I make the point of high negative predictive value, because those that go through, are going to be ones that are going on to confirmatory tests. Those that are on high negative predictive value means they really, really, really are true negatives. If there happens to be some that are missed, they're going to be tested in a year or 2 years or otherwise. So I think if we look at it in that way, as John, to your point, and focus on those aspects for initial test launch, we can get these things in the market, and we can, think about it, even though the biogen demand is, not quite there, Lilly, Roche, Eisai, all coming through, we're going to see a huge influx. And the bottleneck, where do we all go? Primary care physicians with our initial complaint. So having a very basic screening cognitive test and people from an amyloid positivity perspective to push them through, I think can do a lot of good. And then, John, to your point on the misdiagnosis, we've got to embrace other biomarkers at the neurology level for differential diagnosis. Is your tau high? Is your p-tau high? Is your GFAP high? And other aspects. We've got to really address those as well. So I think the key is doing this and targeting high NPV in a responsible way.
Great, John. I guess I'll add a few remaining comments. I think from our early experience in the clinic, I think, one of the initial, to your point, you emphasized, perfection can be enemy of the good. I think in our case, we did spend many, many years, actually over 10 years, working on just amyloid beta 4240. It is a very complex set of analytes to measure in the blood. And I think, there was at some level, a question, you've introduced this test, is the market ready for it? And from our perspective, I think a company that maintains incredibly high integrity and has kind of takes a step back to evaluate the quality of the data, that was the basis for why we introduced the test, because we were highly confident based on those 10 years of experience. So I think, yes, it's important to get these technologies out there, but this point about scalability, I think I really want to underscore some of the points that Mike emphasized, because you need to make sure that you have good analytes. And we clearly are learning that not all analyte tests are created equal. And so I think that the importance of standardization. And once you can standardize your assessments, then thinking about a scalable model. So, from our perspective, I mean, we have a different set of challenges than the immunoassay platforms. I mean, to Roche's credit, they've done a great job with the automation and Alexis. In mass spectrometry, which is our analytical tool that we use to measure amyloid beta 4240 and a whole variety of p-tau related analytes and other markers that will be coming down the road. That first initial focus was on the analytical precision. And that is now paying off substantial dividends because we're starting to see the acceptance of our test in both clinical trials as well as the clinic. So once you've validated your markers and standardized, then the next step is to think about scalability. And that's, we are spending a tremendous amount of time evaluating a variety of different models. I think it's safe to say that from our perspective, we think that quality control is going to be absolutely essential to introducing this test into other regions of the world. And that will be a very significant effort of ours in 2022. We have several R&D projects underway that involve a standardization of our assays that will enable technology transfer and effectively, I mean, I don't want to say seamless because, you're talking about sophisticated technologies, but in a way that will allow us to simply hand over an instruction booklet and with C2N's capabilities and proficiencies, be able to, hold the hands of our collaborators to ensure that proficiency and scalability is there when other labs start performing that test. It's absolutely essential to see scalable solutions that a test performed in one lab is going to give the exact same test result performed in another lab. And so that has to be nailed down before you can start introducing these technologies. And I think, we feel confident that, the robustness of the mass spectrometer-based solutions do allow for that, but it will require distilling down to actionable, concrete instructions that you'll be able to pass on to other labs. One of the things that we did at C2N in the several years leading up to the introduction into the market was the automation of our workflow. So our assay has very high throughput compared to some our academic collaborators lab, Randy Bateman. There was a time when our throughput was about 5 to 8-fold higher, and that represented the industrialization of the assay. So I think that with technological advances and better automation, it's going to get us to that point where we can then really start thinking about transferring technologies to other parts of the world. And that is a key priority for us in 2022.
So, as a patient advocate, we are always pressing for responsible impatience because the situation we are seeing today, as you all have mentioned it, is the global inaccuracy of the diagnostic process, the pain of a false negative or a false positive is really foundationally a source of real human suffering. And in that context, my next questions to all of you are in two parts. Back to the rigor that you all are implying or expressing is obviously critical from a regulatory point of view. Your respective countries, the world more broadly, aren't going to allow tests on them introduced in their respective countries without some level of confidence in their accuracy, sensitivity, specificity, etc. Similarly, though, and Dr. Ip, I'm going to save you to last because China's regulatory environment and the way it allows the distribution of tests is very different. In Europe and in the United States, you have two unique problems that we need to discuss briefly. The first is just people use the United States. Today, if any of you apply to the FDA for a diagnostic approval on your prospective test, you're looking at a 2-year wait. The time is doubled because of the COVID tests. So we have a real backlog. And back to responsible impatience, we need to ask ourselves regulatorily, what can we do to introduce lab-developed tests in all our respective markets to inform physician practice while you folks all march dutifully towards a diagnostic approval in all these markets. So I'm going to start with Sasha to just ask your point of view, and then I'll move around and save Dr. Ip to last as to how do we what's the fastest way to get something responsibly in the market, and what do you see the regulatory process being for Roche?
Yeah, I mean, great question. I think it's quite market dependent as well. You get some variations across Europe. It's difficult to say, but I mean, certainly, you know, we can get assays out there being used in research contexts, and you know, we can be gathering valuable information on how these perform in a real world cohort. Because also the challenge with developing these assays is, you know, we faced it within the CSF. You validate them in extremely well characterized cohorts that are very well selected, and this is not mirroring what we're seeing in real life, and particularly I mean, GP cohorts really just don't exist. So to take into account all the different sort of potential comorbidities, the potential things which might be impacting and causing marker elevations that we don't know about. I mean, these are a lot of the challenges that I think we're facing in taking something from development and bringing it to market. So I think, you know, further research with the assays out in clinical practice, gathering data is a way to get them out there faster, but also to keep building that information, which is essentially, you know, what we need to do. But yeah, I mean, at the end of the day, we need to demonstrate that we can improve standard of care, and it's not hard to improve upon, you know, what we have now. But the challenge, I think, is in most geographies is actually demonstrating that there's a health economic benefit for introducing those tests, because in many healthcare systems you won't get them implemented unless they are reimbursed. So I think, you know, this is the challenge that we'll be facing quite a lot across Europe, and demonstrating that something is better, but also, you know, having that kind of long-term data to say actually it impacts patient outcome. That is, you know, something which, you know, takes time to build and can be quite difficult to demonstrate as well.
Joel?
Yeah, John, thank you. So, you know, I think it's our impression, the Precision AD test is a test that can be ordered today by healthcare providers. So a physician in the United States can order the test for a patient with early mild cognitive impairment or early dementia and doing an evaluation. That ability for the provider and being able to touch patients and offer a solution that is continuing to evolve, but is extremely good in its present form, is made possible through a key engine of innovation within diagnostic testing, which is the laboratory developed test under the CLIA pathway. Okay, so the Center for Medicare and Medicaid Services offers a regulatory pathway, which is really an accreditation of laboratories. When a test is performed out of a single laboratory, the question that comes into play that is certainly of interest to the FDA, but less relevant in terms of regulation oversight, is does the laboratory have the technical proficiency and have they performed the appropriate analytical validation steps to be able to offer that test to the clinical community? And the beautiful thing about that pathway is as long as you have responsible manufacturers and developers of diagnostic tests, meaning they place high value on the integrity of the data, the stringency with which you would determine your acceptability standards, you can introduce the test under CLIA. And in fact, there are many tests out there in the clinic that are current standards of care that are only laboratory developed tests. They are not FDA approved. Examples include like the Oncotype DX test, Genomic Health. Prior to their interactions with the FDA, they introduced the testing as a laboratory developed test. And it was there was the analytical validation, which happens within the lab, but then the clinical validation, and to Sasha's point, demonstrating the clinical effectiveness of the test to be able to improve the patient's outcomes. And so that's the kind of work that's happening at C2N. We're engaged in thousands and thousands of ongoing clinical validity studies in different populations, different races and ethnicities. And we're also involved in extensive clinical utilization studies. I apologize for the phone ring in the background. And so those clinical utility studies are allowing for us to understand how clinicians are going to be able to make better decisions for their patients. That's the evidence package that you really want to drive to enable responsible and efficient adoption. And we're now extending this throughout the world. So we're participating in the Davos collaboration, and we will soon be introducing our test in 6 different regions of the world, which include Brazil, Mexico, Jamaica, Scotland, Japan, and the US. We're starting to broaden that experience to help in these culturally sensitive regions to understand how the test and what it means to convey information to the patients in that clinical setting, because it is so important to respect the cultural differences in how we're going to explain risk or explain the presence of a pathological indicator. So, I think that the FDA is an important regulator to oversee quality, but laboratory developed tests represent another pathway that you can continue to pursue. And they're actually not mutually exclusive. So we have a breakthrough device designation, and we talk with the FDA regularly, and we are working towards that FDA approval. But as you indicated, that can be a lengthy process. And, we're interested in speeding innovation when the technology is ready sooner than that.
Steve and then Mike, and especially what defines success in clinical practice from a getting it regulatorily started.
Right. So echoing everything Joel just said with respect to the laboratory developed test path, and there are good precedents not only for companies that have launched with LDTs in the US, but actually reimbursement. Our partners actually in our CLIA lab have a lung cancer test that they offer that has also received reimbursement. The reimbursement path also equally important in that regard. So it is a valid path. We also longer term are pursuing IVD with kits and next generation device, and it does take some time. But the LDT path seems quite valid for the United States, and that's what we're pursuing. And yes, of course, with all the appropriate analytical and clinical validation, as everyone has described. I think one other point I wanted to make about other markets that you've asked about. In the EU, it is a little bit more straightforward with respect to, there is no classical LDT CLIA lab type pathway, but there are ways to introduce tests there. I think the bigger barrier in the EU is with respect to how those tests are paid for, and to, I think Sasha made the point with respect to the economic drivers as well. It's going to be different in Germany, what that process is, than in the UK non-EU, in the UK with respect to dealing with NHS. We've seen this in ophthalmology where actually you have FDA approved treatments that are not reimbursed until such time as they can show, what the economic impact to that target population is. It's not a gimme at all with respect to those markets. So our approach really is to expand to those markets, not only from a regulatory perspective, but very early on, working through the reimbursement and the right groups that you have to talk to in order to bring forth payment for those tests as well. But I think that, in the US, I think that, there's good precedent for the path that Joel and others and ourselves are pursuing in LDT.
Mike, LDTs, how do you feel about them? I'm excited about it as an initial way to win.
Yeah, so obviously that's the way to start. I think the other speakers have talked about expanding beyond LDTs. There are many regulatory considerations. I'm not going to. I have very little to add there. I want to go to the other issue, though, that several of you brought up in terms of health economics, and I want to go back to my opening statement, and I want to cheat and jump ahead to the call to action, because I remind you, it is unacceptable to have 30% of your patients misdiagnosed when it's avoidable. And health economics is not the issue. The issue is the patient's right to have an early and accurate diagnosis. And those of you in the audience who are in position to make policy, in position to advocate, that's my call to action. Let's stop talking about health economics for diagnosis, because diagnosis will only have a health economics impact when there are, as are coming, disease-modifying therapies. But even in the absence of disease-modifying therapies, diagnosis, correct diagnosis, has an incredible impact on the patient. Economics is not the issue. The issue is going through life being told incorrectly that you have a degenerative disease when you may not. And so that's really where I'd like the policymakers and the advocacy groups in the audience to focus.
That's great, Mike. Thank you. But Dr. Ip, China's very different. And, how do we look at, from a regulatory point of view, laboratory developed test acceptance, what is the current status of the wonderful technologies we're discussing today?
So in mainland China, the equipment and also the reagents for blood biomarkers based diagnosis, it has to go through CFDA for registration and approval based on China regulations on medical equipment and in vitro diagnostic reagents. So this is the process that one has to go through. And as I mentioned earlier, there are two ways to implement it. You can, implement it directly in the diagnostic department of a hospital where the CFDA approved equipment and diagnostic reagents are being used. And then they can run it similar to a routine blood test in the hospital. The other way is to engage a service center for blood tests, and you can engage a third-party service center outside the hospital, and the blood samples can be collected in the hospitals and then sent to the service centers where the CFDA-approved equipment and diagnostic reagents can be used. So I heard recently that the ATN biomarker panel will be implemented soon in mainland China. Having said all that, I would like to point out that based on our study, looking at the blood biomarkers, some are quite consistent across ethnic groups. For example, European descent or non-European descent. Some are very consistent, like NFL and also phosphatide 181. But some biomarkers actually are different across ethnic groups. So I think we need to keep that in mind if we want to use blood-based biomarkers in different countries for different ethnic groups. This need to be validated for different ethnic groups first before we implement the blood tests.
Well, I just want the audience to know I am watching the time. I so appreciate Mike moving us to the call to action. The need for these tests in clinical trials as well as clinical practice is critical. The form and platforms these tests are made available in is also critical. We can't scale, we can't give access to better quality diagnosis if we don't have robust technologies that can be put in rather limited healthcare environments. And that would be my call to action. But real quickly around the horn, what would be your point of view, Dr. Ip, on what the field needs to do next to advance the appropriate diagnosis of AD using blood tests?
Well, I think in developed countries versus the developing countries, the requirement be slightly different. For developed countries, I think the accuracy may not have to be super high because you can use it for a screening tool, and then you can back it up with PET imaging and CSF tests. But for developing countries, I think the accuracy provided by the blood test needs to be quite high in order to better inform the diagnosis.
Thank you. And everyone else, any closing thoughts? We've got 1 minute.
Yeah, John, I'm sorry, go ahead, Sasha.
Thank you. Thank you. Well, the wonderful thing about this meeting, I think, is the participation is so broad, and we've got, representation from clinical practice, policy, academia, industry. And I think it's just this approach which takes really to make a difference. So just to encourage, I guess, everybody listening, continue discussing, challenging the status quo as we've done today. From these conversations, initiatives are born, and, that's how we get action and change ultimately. My panelists and myself are working on the development of these tests. We look to the audience, to those who to help us to implement them into clinical routine. It's going to take education, upskilling, and some radical thinking. So join us on this journey.
Yeah, John, I would just like to say that I think we really need to make a global collective effort towards emphasizing the importance of a precise diagnosis. Because right now, it's the consideration that an individual has dementia is really insufficient based on even the existing tools, let alone future blood-based biomarkers. I think that it does make a difference to patients and their families, and it's highly impactful to ultimately how they plan for the future and how they'll and either take existing therapies or in the very near future, disease-modifying therapies that will have a profound impact. So we need to start gravitating towards using these tools and get away from a general impression of an individual with dementia without the ability to do that much. I think this is going to be a very significant effort that PCPs, specialists, and patients in general are going to have to embrace, and something we should be helping to support.
Steve, and then Mike, you finish. We have time for both of you.
Okay. So I actually echo I'm not going to repeat everything Joel just said, because I actually agree 100%. That's what I would have said. I think I like to look at things from a critical path perspective and where we are today. You said, what's actionable today? How can we move the needle? We're going to see a tsunami of new treatments coming through in the next couple of years. We're going to have bottlenecks in the healthcare system, even in the United States, where we have access to certain tests, with an influx at the primary care level where people go. I think one of the things that can be very helpful in moving the needle, and if anyone from CMS is listening, is to really embrace blood biomarkers in a way that is responsible, as I pointed out before, with high negative predictive value, to start to deal with this influx of patients to gate them into neurology. I think that is absolutely imperative that they address that in their January memo and their decision upcoming next year for Aduhelm, which will have implications broadly for the other disease-modifying therapies, taking a position on how blood biomarkers will play a role in that. In the absence of that, it'll be up to payers, because FDA shifted a lot of the responsibility of decision-making onto payers. So this decision for Aduhelm is critical, and we'd like to see blood biomarkers be addressed. Some companies are, much farther along than others and have done a really great responsible job, like Joel at C2N, that I think is well positioned in all the work that's been done to be positioned for this sort of thing. So I'd like that when I look at, issues of how do you really gain traction? What are the critical path elements? That's really right here and now. Everything longer term, I agree with what Mike said and what Joel said in responsible diagnosis. But where we are today with, we don't get to choose on the timing of when these drugs come through and when the demand is there. So we've got to have something that can responsibly gate these patients through. And I'd like to see and I'm talking US centric because it'll set then the tone for other markets. So I'd like to, see that addressed in January through April from CMS when they're looking at this very important problem.
Really good point. And Mike.
So again, there's no doubt that molecular biomarkers are going to be essential. You can't treat with an anti-amyloid therapy if the patient doesn't have amyloid. So we've got to have ways to evaluate that. And I think our commitment with the tens of thousands of patients we've studied in trials and collaborations we have around the world is really to understand how the various approaches fit together best, what's the best algorithm, which assays and when can you use them alone, when do they have to be backed up by PET or some other analyte or some other diagnostic tool. So on the one hand, strongly support everything everybody said. We want to have payers and regulators approve these things as soon as they are appropriately validated. And on the other hand, we will continue the investigations to find the best utilization, the best algorithms, because that's what we in the field should be doing.
Well, I want to thank you. We could have done another hour easily, but I want to thank the panel. You've been terrific. I think the nuances of these emerging technologies are now fully in front of our audience. And I just want to finish by saying these need to be deployed responsibly, quickly, rigorously, but in scale for the good of patients with AD or other neurological disease. And I thank you all for your labors in this area. And I thank back to you, George.
Thank you, John. Thanks for this really quite excellent discussion, not just upon the status of the science, but on the important issues about how to get these into the marketplace and to get them into the marketplace at scale. It's clear that there's a sense of urgency, but also a sense of pragmatism of assuring that we get things into the marketplace which are accurate and do not give us false positives and negatives. So I really appreciate the really sophisticated discussion that you just moderated. So that will obviously be useful for clinical trials. It's going to be useful as well for clinical practice. And it's now time for a break. We'll see you back here at 10:25 Eastern, 16:25 Europe. When we come back, we're now going to hear, or then you're going to hear from Phyllis Barkman-Farrell of Lilly and the Davos Alzheimer's Collaborative on how we can take the innovations in detection and diagnosis and integrate them into clinical care around the world. Take care. See you at 10:25. Bye-bye.